Good day, everyone. I'm Kara Witcoff, Global Head of Patient Engagement at AskBio, and I'm delighted to be here today. We appreciate the opportunity to partner with Defeat MSA Alliance on this educational program. AskBio is a leader in the field of cell and gene therapies and is now a cornerstone of Bayer's Cell and Gene Therapy Unit. Now to my clinical development colleague, Dr. Akash Pandhare, to share some of our ongoing research. Akash?
Thank you, Kiara. Hello, everyone. My name is Dr. Akash Pandhare. I am the clinical lead for the MSA study at AskBio, and I'm really delighted to be here today to share some of our ongoing research. AskBio is a leader in the field of cell and gene therapy and is now a cornerstone of Bayer's Cell and Gene Therapy Unit. So here are my relevant disclosures. So today, we will be introducing you to the Regenerate MSA 101 trial, a phase 1 clinical trial of AB1005, a gene therapy for the treatment of multiple system atrophy, or MSA. During this presentation, we will cover how this investigational gene therapy works, how the clinical trial is structured, and how to get involved if you are interested in participating in the trial. So MSA is a rare, fast-progressing neurodegenerative disease that is caused by a buildup of a specific protein in certain area of the brain. MSA is characterized by a disruption of processes that the body usually does automatically, such as controlling bladder function or regulating blood pressure. Because of this, individuals with MSA may experience bladder control issues or dizziness when standing up. So MSA is characterized into two major subtypes based on the type of movement symptoms that patients experience, in addition to the disruption of automatic processes. Patients diagnosed with the cerebellar subtype of MSA, or MSA-C, may experience a number of symptoms that result in a lack of coordination or impaired speech. Together, these symptoms are collectively known as cerebellar syndrome. Patients diagnosed with the Parkinsonian subtype of MSA, also known as MSA-P, may experience the same movement symptoms as patients with Parkinson's disease, including tremors, rigid muscles, and slow movement. Some patients with MSA have movement symptoms from both subtypes. The symptoms I mentioned are not the only symptoms a MS patient with MS may experience, but they are the main symptoms used for diagnosis and characterizing MSA. The three major groups of symptoms seen in patients with MSA are related to the parts of the brain that are most impacted by MSA. Disruption of automatic body processes means the neurons in the brainstem are not healthy or functioning properly. Cerebellar syndrome means neurons in the cerebellum are under stress and not able to function. And Parkinsonian symptoms indicate neurons in the parts of the brain related to movement are not functioning properly. Neurons, which are specialized cells in the brain, are kind of like people in the sense that neurons need connection and communication to survive and thrive. The neurons in these different parts of the brain, the brainstem, cerebellum, and putamen, are connected in a large network. Dysfunction in one part can spread to other parts as the connections start to deteriorate. AskBio's investigational gene therapy, AB1005, is being evaluated for patients with MSA-P. So let's take a closer look at the processes behind the Parkinsonian movement symptoms. The movement symptoms in MSA-P and Parkinson's disease are driven by lower levels of chemical in the brain called dopamine. Dopamine is a key messenger in the parts of the brain that control movement, such as the putamen. In MSA-P and Parkinson's disease, the neurons that control movement by using dopamine to communicate are not able to function properly and eventually die. Dopamine replacement therapy can help manage the movement symptoms of MSA-P, but it does not fix the underlying disease. As MSA-P progresses, the effectiveness of the limited options available decreases. Currently, there is no cure or treatment to slow down MSA. So gene therapy offers the potential for treating neurodegenerative disorders such as MSA. Genes are segments of DNA that act as the blueprint for what proteins the cells in our body should make, and gene therapy harnesses this process to make proteins that can act as the medication. So gene therapy works by delivering genetic material into a patient's cells to treat the causes of disease. And there are different ways that gene therapy can work to help people, including introducing an active copy of a gene into the body to help treat a disease, also called gene transfer therapy, turning off a bad gene that is causing a disease, also called gene silencing therapy, replacing a bad gene with a healthy copy of the gene, also known as gene editing therapy. So today, we are going to focus on gene transfer therapy, which is how AB1005 works. So in gene transfer therapy, the genetic material is packaged in a carrier called a vector, which ensures delivery to the proper cells. And if you think of the genetic material as a ladder, then the vector is an envelope with the address to ensure delivery. Excuse me, I just made a mistake. To ensure the delivery to the proper cells. If you think of the genetic material as a ladder, then the vector is an envelope and it ensures its delivery to the right location. Vectors for gene therapy are commonly made from adeno-associated virus, or AAV for short, but these viruses have been altered and will not make you sick. Scientists use viral vectors to deliver gene therapy because viruses are very good at getting inside cells. Since the genetic material of a gene therapy needs to get inside the patient's cells to be effective, viral vectors are the ideal envelope for delivering gene transfer therapy. So the vector used for AB1005 is the adeno-associated virus type 2, or AAV2. So to sum it up, a gene or letter is placed in an envelope to create a package, the gene therapy, which is then delivered to where it needs to be in the body, so the patient's cells can use that information to make a protein that acts as a medication. So gene therapy could be used to preserve or restore the function of brain cells that are lost as MSA progresses. So researchers have found a loss of important growth factors needed to promote healthy function of brain cells in patients with MSA. The lack of these growth factors, such as glial cell line derived neurotrophic factor, or GDNF, may contribute to MSA disease progression. GDNF is especially important for the health of those neurons in the putamen that use dopamine to communicate to control movement. The genetic material in AB1005 is a copy of the gene to produce GDNF. So AB1005 is delivered directly to the putamen, the shape shown in the middle of the brain, through a minimally invasive MRI-monitored one-time brain surgery. The specific administration technique used allows the gene therapy to specifically target the structure. The percentage of target neurons that are able to take in the gene therapy is thought to be important for efficacy. So achieving sufficient coverage of the putamen is key. Any type of brain surgery is scary, but administration of AB1005 to the putamen is comparatively less invasive than other neurosurgical procedures, and there is no hardware left in the brain and typically no need for additional surgeries or maintenance. This is a one-time treatment that is expected to be long-lasting. Here, we have an animation showing what the administration would look like. So to summarize, GDNF is important for the health of neurons that use dopamine to communicate to control movement. Loss of GDNF and deterioration of the neurons lead to the movement symptoms seen in patients with MSA and Parkinson's disease. AB1005 is a gene transfer therapy made of an AAV2 vector that carries a copy of the gene to produce GDNF. By providing an extra copy of the GDNF gene to increase the amount of this important growth factor directly in the putamen, AB1005 may potentially support the health and survival of these neurons, which may slow disease progression. AskBio is currently recruiting patients for a phase 1 clinical trial to evaluate the safety of AB1005 and better understand the biological indicators of MSA. This study, known as Regenerate MSA 101, is a randomized, double-blind, controlled trial that consists of a one-time brain surgery to administer AB1005. In this trial, patients will be randomized in a 2:1 ratio to receive AB1005 or a controlled surgery with no active treatment. This means that 2/3 of the patients enrolled in Regenerate MSA 101 will receive AB1005 and 1/3 of patients will be the control group. Control groups are an incredibly important part of clinical trials and are needed to evaluate if the treatment is working. Eligible patients for this trial are those who are within the age group of 35 to 75 years, have been diagnosed with MSAP, have the ability to walk with or without an assistive device, have not had prior brain surgery. Enrolled patients will be required to visit the study center approximately 5 times over the first 12 months and then twice a year for up to 3 years. During these visits to the study center, patients will undergo physical, neurological, and blood tests. In addition, patients will be asked to complete at-home questionnaires about their health and quality of life. Patients who participate in clinical trials help scientists and physicians better understand MSA and how to improve it with treatment. Trial participation contributes to the advancement of the medical field and may make a positive impact on the lives of people affected by MSA. For more information on Regenerate MSA 101, please contact askfirst@askbio.com or scan the QR code on the slide. Thank you for taking the time to learn about AB1005 and the Regenerate MSA 101 study. Now, I'm back to my colleague Kara, who will provide more information on AskBio and the AskFirst patient engagement program.
Thank you, Akash. Working at AskBio means working at the forefront of gene therapy. AskBio has had many firsts, including being the first to clone an AAV vector for therapeutic purposes and the first to deliver an AAV vector to the brain. AskBio is committed to exploring potentially disease-modifying gene therapies in MSA and other difficult-to-treat diseases. Across all therapeutic areas, we take pride in our patient-centric approach that focuses on collaborative patient advocacy. AskFirst is our collaborative program for advocacy groups, patients, and their families who may benefit from the latest advances in gene therapy research and potential curative therapies. We welcome the voices of patients and their families to find answers for advancing therapeutics, technology, regulatory requirements, and public policy. Thank you to Defeat MSA Alliance for including us in this event, and thank you to all who attended today. If you have any questions on what you learned, or if you're interested in participating in Regenerate MSA 101, please contact AskFirst@askbio.com or scan the QR code on the slide.